Okay, so welcome to Shanghai from me as well, to this inaugural conference, which we felt was kind of time appropriate, as I think many of you are now developing very innovative new medicines or molecules that could be quite successful in the market in China and abroad. And we feel that there is a significant interest potentially in this area, and a lot of you are still evaluating, how you should maybe undertake some of the work and also understand the rationale for investing in this particular area. So I will talk a little bit about the commercial rationale for why you should be spending money on these kind of assays and this type of work. So I think it's been touched on by the other speakers that basically it's a question of competitiveness as well. So immunogenicity risk is often only considered as a sort of regulatory question, or that's how a lot of companies in Western countries look at it. In reality, it really, as Edmund has mentioned, and also Tim, it determines the market competitiveness of drugs and of the sort of drug development and manufacturing platforms that you are pursuing. And really, one of the unfortunately worst outcomes that you could potentially have is that you actually successfully develop your drug, and then you actually fail in the marketplace because this drug misses its potential due to immunogenicity. And Tim and I were talking earlier that actually there is a graveyard of failed drugs due to immunogenicity, and you don't hear much about it because the developers don't like to talk about it very much, but it does exist. There are a few case studies that are worth mentioning. The Vitreptocog was certainly one of them, and I think there will be potentially another one to be talked about in this meeting. So the point is that it doesn't matter what you do. Anything that you do as an intervention can be, and in reality actually will be, the cause of an immune response. That's essentially unavoidable. So the question from a business perspective is really whether the level and nature of this response in the context of what you're trying to achieve and what you're trying to treat and the competition that you'll be facing is acceptable to the end user or the people who pay for the drug in the marketplace. So most developers, if they're larger companies, have a mix of drugs in their development pipeline and a mix of approaches. And typically with larger companies, we see that, of course, they are pursuing a mixture of existing mechanisms of action with generics or biosimilars, and also novel mode of action and novel platforms. So each case is a bit in its own category as regards immunogenicity. Clearly, where you have an existing mode of action and you're sort of competing with other drugs that are doing similar things in the marketplace, it's not really that mode of action that will distinguish you in the marketplace. It'll be other product attributes. And we think that immunogenicity is really one of those key attributes that can make a huge difference here. And if you're at a disadvantage, then that could be a major drawback. And if your profile is better than the competition, that could be a significant advantage. Of course, if you have a new mode of action that no one else has put a drug out for, or it's maybe a novel type of compound, or even an entire platform that's new and not so much is known about it, then there will be significant unknowns of how this intervention will interact with the immune system, and you may even have systematic immunogenicity risk. What that means is that your entire platform could be affected by a problem if you don't catch it out early. And even a low percentage of treatment failure can contribute to high additional overall costs to payers. So that's particularly true, as an example, in the factor replacement market for hemophilia, where factor VIII is typically given as a first line. So it's a very expensive intervention, costing typically $300,000 a year, last time I checked, and it may have gone up since then. And a significant number of patients basically develop anti-drug antibodies against this factor VIII there is a way of dealing with it in the first instance, just give more drug, right? So what if you just overwhelm the immune system, just add more drug until the patient still responds? But that can push the treatment cost into the low millions per year in that case. So it's extremely expensive. So there has been a huge drive to reduce the immunogenicity of recombinant factor VIII proteins for that reason. So you've already seen the Treptococcus study, so I won't dwell on it too much other than to say that, or reflect on the fact that yes, immunogenicity was really the root cause of failure for this drug. It was the rather low numbers of mutations that were introduced that Novo Nordisk felt would be, a significant advantage in this product by making it more active. Unfortunately, it basically killed the drug at the end of the day. And the problems were only found in phase 3 trials. So even in phase 1 and 2, you didn't see these problems. But what we could see from the postmortems or after this drug had been killed, studies that we and Novo together had undertaken, is that the preclinical assessment tools were capable of actually finding these issues and reflecting them appropriately. So just coming back to T-cell responses again and their relevance, you've seen the introductions in terms of the mechanistic picture on the previous slides. I just want to reiterate that differentiated drug antibody responses, so the ones that are high affinity, really do need T-cell help to mature. And a protein that doesn't have this T-cell help simply isn't going to be a very strong immunogen. And there really isn't a doubt about this point, I would just reiterate. So we know this from basic science in general, immunology. It's a proven fact, and it's also very much borne out in vaccine science. And the good news is that we are really, with the assays that Edmund has been talking about, able to understand human T-cell antigenicity of a drug without putting this drug into people first. And that's possible through physical in vitro assays, which we and others can carry out. So again, another small example here is, what happens if you actually humanize an antibody. This was a project from some time ago where it was actually a biodefense project against a potential bioweapon where the antibody was directed at Venezuelan equine encephalitis virus. And it wasn't possible to actually test this in human trials at the end of the day because this is not a disease that people would typically get. So the sponsor wanted to basically humanize this antibody and demonstrate that it would reduce immune responses should it ever be necessary to use this drug. And you can see how going from the chimeric to the humanized version in the peptide T-cell ProMap assay, the responses to peptides are significantly reduced. So this was then viewed as a success in the humanization approach. So there it is. So we've touched on Humira as well. And I just want to reflect on something here. Edmund has talked about the case study. We talked to developers here in China, and the responses that we're getting or the interactions sort of remind us a little bit of what we used to hear in the US and Europe maybe 10, 15 years ago, which is, in the case of a drug like this, which is a fully human drug based on phage display, the idea was, there isn't any immunogenicity to this drug. That's what people thought back in 2008 to 2010. And similar sort of drugs that were sort of a fully human origin. So the label for Humira actually did already indicate that there would be ADAs between 1 and 12%, but in post-marketing studies, the incidence has been demonstrated to be much higher, and that's maybe also because the clinical trial environment doesn't really reflect how the drug is used in patient populations downstream, where the control is a lot less, especially with a drug that people administer themselves, which is true of Humira. So Edmund touched on the ProPresent results. So we could clearly identify some presented sequences that won't show the T-cell assay data again, which then were also validated in T-cell assays. So it was possible to see clearly that there were issues coming from the CDRs of these molecules, which even in a fully human antibody are always sequences that are never in your patient as naturally present sequences. So the fact that it's derived from a fully human library doesn't matter anymore in that nature. So when we then look at what people are trying to develop, we can see, and this has been commented on in the sort of conference circuit as well, that even standard antibodies already often hit immunogenicity hurdles, and some of the examples that have been presented reflect this as well. And then, there's a whole sort of plethora of new approaches that people are now are pursuing, which are all exciting, hold a lot of potential. But as you basically get more sophisticated in how drugs are designed, then the potential issues also become more complex and challenging. So in antibody-drug conjugates, we know that conjugation can, first of all, cause the hapten of the conjugate to be presented. And secondly, there can also be a fundamental change in the pattern of antigen presentation. So when your drug gets taken up by APCs, this can change basically how the enzymes break down the drug and therefore present peptides as compared to the unconjugated drug. So that means that germline sequences that are you're sort of theoretically tolerant to, that would normally never be presented, can be presented, and therefore they become immunogenic. We've seen the CRISPR-Cas study. CRISPR-Cas holds an exciting promise, but those proteins are completely foreign, and whatever you're introducing as a change genetically is also potentially new and foreign. With viruses, you have several issues. You potentially deliver immunogenic genes, capsid proteins, and you may even have pre-existing immune responses to those viruses. CAR-T and TCR T-cell therapies have specific challenges. They are potent immune stimulators, typically, and also, their job is to be particularly long-lived, so they don't, sort of phase out or get sort of metabolized like a normal drug. So if you do have an unwanted response, it may be very difficult to actually switch it off. And there are some very bad examples where that's led to people dying in clinical trials. RNA LNPs, I think, are very exciting in terms of their potential promise for developing exciting new drugs in the future. But almost all elements in these vehicles are potentially immunogenic, starting from the oligos themselves, the gene products, the targeting proteins, the encapsulating moieties, and so forth. So there's a huge amount of information to be gathered to understand how RNA LNPs really interact with the immune system. And I don't think we've really begun to scratch the surface there. We've touched on synthetic peptides, which can potentially contain stimulatory impurities, and expressed drugs in general have the scope for wholesale protein issues, protein packing problems. So your cell line, which isn't human, is going to translate and pack your protein slightly differently than it happens in a human. So the FDA has actually done some work on showing, and also with analysis performed by us, showing that that's relevant in immunogenicity to recombinant factor replacement proteins. So it's a very interesting and new field of science. Glycosylation, variation, aggregation, and there's many more factors that play a role as well. Coming to the financial part. So just looking, in terms of the finance, what difference does all of this make? It's worth looking at how large companies work financially, which is maybe where many of these programs eventually get licensed into or developed internally end up. So when I looked at this last at the end of March, Eli Lilly was worth nearly $900 billion, Roche a $250 billion Swiss francs, and Pfizer $160 billion. Each of these companies, sort of on average between them, they spend about 20% of their revenue, which is 60 billion across the board, except Roche's is in francs, the other two are in dollars. That's their revenue. They spend 19% or 20% roundabout on R&D, and typically 20% of their revenue drops to the bottom line. And the other important factor is that the PE multiples that we're seeing in the market are averaging at about 26. And interestingly, that hasn't changed too much over the last 15 years. So that tends to be a fairly stable dynamic of, the sort of money that's being made, what people spend on R&D, and what they expect to earn as a consequence, and how that translates into company value. So if you consider a model case study, a company that maybe turns over $10 billion in revenue, and it spends, 20% of its revenue on R&D, that's $2 billion. Its profit after tax is also 20%, so another $2 billion, and the price earning multiples is about 20. Well, that company would be worth $40 billion by multiplying out those two figures. And their R&D investment, the 2 billion, would be expected to replace its revenue maybe every 15 years or so, and hopefully add some growth as well and keep up with inflation as in addition. So if you now consider, the value of investing in immunogenicity risk management throughout the development stages. So we estimate that the cost of a comprehensive program is maybe $600,000 per year per drug program, or if you have maybe 20 different programs in your pipeline, then that could add up to, $12 million. So over 15 years, that would be a considerable spend of 180 million. So it is not inconsequential. What difference would it make? Well, we think there can be a material difference in competitiveness from doing this, and even small aspects of competitiveness can easily increase your revenue by 20%, or if you're less competitive, decrease it by, let's say, 20%. So if you can get that pickup of maybe 20% in revenue, that means that your $10 billion company becomes a $12 billion company. And you will not really require, if you've done this ahead of time, to do more R&D to achieve this. And equally, there aren't any other costs that are at play. There isn't much additional overhead. So much of this should drop to the bottom line. So even if only 50% drop to the bottom line, that would mean you may make another $1 billion in net profit. So on a graphical representation, sure the pointer, you can't see it. So your profit and loss beforehand, where you were turning over 10 billion, you were making 2, your company is worth 40. Afterwards, you're making 12 in revenue and 3 in profit, but you get a multiplier of 20. Oh, that's the point. That's the more powerful one. You make a profit of 3, and that additional profit multiplies by 20 as well. So your company worth 40 billion. Sorry, it becomes a company worth 60 billion. So it lifts your company value by 50%. And when you think about it, I could be slightly wrong about this figure. Maybe I'm also slightly wrong about the 20 billion. But this is 100-fold excess. And even if there are some significant estimation errors, by putting this proposition forward, there's still a very, very, very good chance that you'll be very right on making this investment. So the other thing I would say, and this is just a reflection of what we've seen over the years in other markets that we're active in over the last 20 plus years, is that in those other developed economies around the world, since the late 2000s, immunogenicity risk management using physical bioassays has been adopted by pretty much all major drug developers outside China. There's, by now, I would argue, also a community consensus in these countries that this approach prevents drug failures and loss of competitiveness in the marketplace. So The insights that you gain with these sorts of programs is you can use them to deimmunize drugs, as we've just heard, or select those drugs with the best profile, including based on sequence, physiochemistry, formulation, manufacturing methods, impurity composition, fill finish, and many others. The sort of post-mortem studies that people have undertaken, including with us, where drugs have failed due to immunogenicity, show that the failure does correlate with T-cell activation and antigen presentation and HLA binding assays. So the assays, in a sense, validate this question ex post. And logically, when you think about it, you do a lot of physical assays for other purposes in putting drugs forward for development. Using physical assays to support protein design can only lead to better design choices and with improved outcomes as they cover significant additional physiological depth compared to predictive in silico methods. So in summary, immunogenicity is a product attribute that can be managed, so you can get on top of it. Managing this attribute, we would argue, is not optional, but it's essential for ensuring that a drug reaches the market with a competitive advantage, or at least not with a significant disadvantage. So patients, physicians, and payers will choose the best product available to them. That's only logical. The immunogenicity profile will be one of these key attributes that will be under consideration downstream. So it shouldn't be a piecemeal effort to invest in immunogenicity risk management, however you're planning to potentially do it. And I would very strongly argue that you need a coherent strategy that you need to deploy for your entire product pipeline throughout all product development. So somehow you need to have an idea of how you're going to cover your risk assessment. And that will, doing it on a system basis, will help you also gain very significant cost efficiencies and the best value gain for making the right choices at the right time. If you're asking questions in isolation, it can be confusing and also it can seem expensive. But when you do it for all your efforts together, you gain a lot of synergy in terms of the data that you're pulling out and that you can reuse over time. So the potential return on investment and the margin of safety this can provide against estimation errors should make the decision to manage immunogenicity risk as a core company focus compelling. And with that, I'll end. And if you have any questions, please ask them.
Thank you, Nick. Just probably a comment on the costs. So I think the way which in a typical development program we look at, we're still spreading that cost over quite a period. So it may actually be less than you're talking about here per program.
Yeah.
The other thing is I've worked on a peptide program where we were looking at potential costs of the ADA assays and the NAB assays. And for a single program for a phase 3 study, we could project the difference between a higher immunogenicity molecule and a lower immunogenicity molecule was $10 million just in testing.
Wow.
And
Okay.
so you could pay for a whole year of early immunogenicity assessments by knowing that one product you take forward was of lower risk. Maybe another comment you might also want to talk to. So you've done a projection of costs, and we know in our organisations that these costs get projected and they have to be because they have to be done. And we also then project the immunogenicity. And I think we're now probably better at predicting immunogenicity than we are at costs. So it may be another compelling reason to do things, but I know probably from a ProImmune side as well, you look at your own business costs. Maybe, what do you think? Are we at least as good at predicting immunogenicity as costs for programs?
I don't know. I mean, I think that's probably different for each company. Yeah. I mean, theoretically, predicting the financial side should be easier, but yeah.